Dr. Jennifer Doudna was born in Washington, D.C., and then her family moved to Michigan and then again to Hawaii to support her father's career as a researcher and a collegiate professor. As she explains in an interview with the Noble Foundation, growing up she felt very isolated on the island of Hawaii as she was a non-native. This feeling of being left out made her more driven to prove herself academically. Upon learning about Rosalind Franklin, a pioneering female scientist crucial in the discovery of the structure of DNA, she decided to pursue science despite her advisor's guidance not to. She then pursued her education at Pomona College and then at Harvard Medical School. As Dr. Doudna explains herself, she enjoys gardening and, quote, working with her hands. She says that being so hands-on is what drew her to science in the first place, and she works to grow plants native to her childhood home of Hawaii in California, where she now lives, which she shared in a 2014 interview with the National Institute of General Medical Sciences. I personally admire Dr. Doudna because of her intense dedication to the field of biology. She has made discoveries that are now foundational to our knowledge and understanding of genetics. I also admire Dr. Doudna because she was never deterred by other people's stereotypical understandings of who she was and what she could be within society. So, Dr. Doudna researches in genetics, and before utilizing the CRISPR-Cas9 gene-protein complex from bacteria for genetic editing, her primary research was surrounding RNA. So most of her undergrad, as she details in her interview with the Noble Foundation, was actually focused on RNA, and the most of her doctorate work was also focused on RNA within cell complexes. So Dr. Doudna actually explains the stumbling upon of the CRISPR-Cas9 protein complex as a happy accident. So CRISPR-Cas9 is arguably her largest and most important discovery, and it is found in nature as a bacterial defense against viral DNA, where the Cas9 protein destroys viral DNA before it can infect the cell. Dr. Doudna and her colleague Dr. Emmanuel Charpentier pioneered the use of this function to genetically modify DNA with cells using Cas9 as, quote, DNA scissors. So in her recent paper titled Rapid DNA Unwinding Accelerates Genome Editing by Engineered CRISPR-Cas9, Dr. Doudna and her lab team investigated the difference in efficiency between the GeoCas9 protein and the mutated iGeoCas9 protein when they were used for gene editing in mammalian cells. So the figure now shown on screen is a summary figure representing the experiment as a whole. GeoCas9 was predicted to be very effective in mammalian cells at working as part of the CRISPR-Cas complex. However, it actually had poor levels of activity. The variant iGeoCas9 protein was created, and this protein has mutations along the wedge domain, which allow it to increase activity but still remain stable within the cell. So the experiment used cryo-electron microscopy and biochemical analysis to compare the structure and function of the wild-type GeoCas9 and the variant iGeoCas9 protein. Both the variant and wild-type protein were tested using the same biochemical analysis. They were used to edit the same genes within the same mammalian cells under the same in vitro conditions. So there were many controls in this experiment. And it was found that the iGeoCas9 protein was more effective at editing the mammalian cells because of the wedge domain mutations, which are notated at the top of the figure. These led to new interactions in the PAM region, and this led to increased R group formation, which you can see if you look down the figure. This increased R group formation allowed for better DNA duplex formation. So essentially, the iGeoCas9 protein, the mutated type of the protein, was more effective at gene editing within the CRISPR-Cas9 complex in mammalian cells. So, as I mentioned, Dr. Doudna's largest discovery so far is that of the CRISPR-Cas9 gene editing technique, a technique now widely used since its invention in 2012. As for future directions for her research, she details on her lab website in the structure section that she is going to continue looking at the CRISPR-Cas9 system within E. coli and the effects of other Cas proteins within the system, which is what the paper I just explained was looking at. Doudna's continued research into CRISPR-Cas will continue to further our understanding of gene editing techniques, and it is incredibly ironic and amazing that this just happened to be a discovery she stumbled across. So thank you for watching and learning about Dr. Jennifer Doudna with me. I hope you enjoyed.